Welcome back to my channel Knowledge Chronicle.
Did you know your body copies over 6 billion letters of DNA every time a single cell divides? And it does it with near-perfect accuracy? But how exactly does DNA replicate? What makes this process so precise, and why did Watson and Crick's discovery change everything? Let's unravel the mystery of life's ultimate photocopy machine, DNA replication. Welcome to Knowledge Chronicle, where science meets curiosity. In 1953, James Watson and Francis Crick proposed the double helical structure of DNA. But what made their discovery legendary wasn't just the structure, it was the implication. They said, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." In simple terms, they realized DNA could make a perfect copy of itself. This laid the foundation of what we now call semiconservative replication. So what does that mean? Imagine a zipper unzipping. Each half becomes a mold to make a brand new matching side. The result? 2 DNA molecules, each with one old and one new strand. That's semiconservative replication. Let's fast forward to 1958. Two scientists, Matthew Meselson and Franklin Stahl, put this theory to the test using Escherichia coli, a bacteria. How did they do it? A. They grew E. coli in a special medium where the only nitrogen available was 15N, a heavier isotope of nitrogen. This heavy nitrogen got incorporated into the DNA, making it denser. Then, they transferred the bacteria into a new medium with normal nitrogen, 14N, and allowed them to grow and divide. At different time intervals, after 20 minutes, after 40 minutes and more, they extracted the DNA and ran it through a cesium chloride density gradient using centrifugation. Quick side note, centrifugal force is like spinning clothes in a washing machine. Heavier stuff moves outward faster. So, heavier DNA sinks lower than lighter DNA. What did they find? After one generation, 20 minutes, all DNA had an intermediate density, proving it was a mix, one old strand with 15N and one new with 14N. After 2 generations, 40 minutes, half the DNA was light, 14N, and half was hybrid, 15N. This confirmed semi-conservative replication. Similar experiments on faba beans, Vicia faba, using radioactive thymidine gave the same results in plant chromosomes. DNA replicates the same way, even in humans. Replication isn't just about unzipping and copying, it's a carefully orchestrated biochemical ballet run by molecular machines. Let's explore. The STAR enzyme, DNA-dependent DNA polymerase. This enzyme reads one strand and builds the new complementary one. It's called DNA-dependent because it needs a DNA template, and DNA polymerase because it strings together DNA building blocks. But here's the insane part. In E. coli, this enzyme adds about 2,000 base pairs per second and finishes the whole genome, over 4.6 million base pairs, in just 18 minutes. Now imagine that happening in every bacterial cell constantly. It's speed and precision in one. So what are the raw materials? DNA is made of nucleotides, A, T, G, and C. In replication, deoxyribonucleoside triphosphates are used. They serve 2 roles, first as building blocks and second as energy sources. Like ATP, they release energy when their high-energy phosphate bonds break. The replication fork. DNA can't unzip all at once. It's too long and would need too much energy. Instead, it opens up at small sections called replication forks. Here's the twist. DNA strands are anti-parallel. One goes 5' to 3' and the other 3' to 5'. But DNA polymerase only works from 5' to 3', so replication happens differently on each strand. The leading strand is replicated continuously. The lagging strand is replicated in pieces, called Okazaki fragments. DNA ligase, the molecular glue. These fragments aren't useful unless joined together. That's where DNA ligase comes in. It stitches the Okazaki fragments into one seamless strand. Origin of replication. Replication doesn't start just anywhere. There's a fixed starting point on the DNA called the origin of replication. In bacteria like E. coli, there's usually one. In humans, there are thousands. That's how our huge genome gets copied on time. This concept is also crucial in recombinant DNA technology. When scientists want to copy a piece of DNA, they use vectors like plasmids that contain an origin of replication. Replication in eukaryotes. In higher organisms like us, Replication happens during a specific phase of the cell cycle called the S phase, short for synthesis phase. This ensures that when the cell divides, each daughter cell gets a complete copy of the DNA. If something goes wrong here, say the cell copies DNA but doesn't divide, problems like polyploidy can happen. That means having extra sets of chromosomes, which can lead to developmental issues or cancer. And although we've uncovered much about replication, many details in eukaryotic cells remain under research. Let's tie it all together with a relatable example. Imagine baking a cake using a recipe. The original recipe book is your parental DNA. You open the book unzipping the helix, copy each line using polymerase, and now you have two books. Each has one original page and one newly written page. That's semiconservative replication. And to make this cake at lightning speed, you have helpers, like helicase to unwind the book primase to start the copying, polymerase to do the writing, and ligase to bind the pieces together. DNA replication is the foundation of life. It allows your cells to divide, your body to grow, and your genetic legacy to be passed on. From bacteria to humans, this beautiful process stays the same, precise, regulated, and mind-blowingly efficient. Next time you get a paper cut, remember, millions of cells are replicating their DNA to heal you right now. If this video helped you understand how life replicates itself, give it a thumbs up. Your one like, share, and subscribe will help us create more valuable content. Stay curious. This is Knowledge Chronicle.